So Preeti, when you talk, you on your mic, you on your video.
Okay, sir. Sure.
Okay.
Done, sir.
I believe I'm visible, sir.
Okay, then we can start now.
Okay.
Today
I believe
we
I'm
have
audible.
1 minute. Let me say one or two lines about Madam.
Sure.
Today we have a very accomplished professor and scientist, Professor Dr. Anita Pandey. I know her since many, many years, and she is a very sincere and dedicated researcher. We are very fortunate to have her, and she will speak on microbial diversity. So, Supriti, you can introduce.
Okay, sure, sir. So good evening, everyone, and thanks for joining. My name is Supriti Bhalla, and I'm a member of MBSI. I appreciate everyone joining the meeting for talk today. I'll be starting with a brief introduction. The Microbiology Society of India was established with the aim to encourage and support research in microbiology, along with cooperating and affiliating with international organizations in field of microbiology in 1996. The society has many renowned microbiologists, teachers, researchers, students, and life members. I would like to welcome the founder and president of the Central Council of MBSI, Dr. Arvind Deshmukhji, a former professor and the head of microbiology department at Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He taught classes in the field of microbiology at the postgraduate and graduate level and teaches introductory microbiology. Beyond his academic work, he is a writer and a keynote speaker. I would also like to welcome the president of Tri-City Group and also the former head of environmental biotechnology and microbial biochemistry at CSIR IMTech, Dr. Saranjit Singh Ji, sir. The name listed in top 2% of global scientists for 3 consecutive years. He has served in MTCC and Gene Bank, which is now the first IDA in India. Been a member of Task Force, National Biodiversity Authority Chennai, and 2 WFCC committees. He has also been awarded the Loyola Environment Award, Lifetime Achievement Award 2022 by St. Xavier's College, Ranchi, and Lifetime Achievement Award 2023 by Shalini University, Solon. His publications are in the High Impact Factor Journal and articles are highly cited in the literature with over 18,200 citations, H-index 58 and I-20 index 150. Dr. Saranjit Ji makes very sincere efforts and works very hard for inviting eminent scientists to the show from diverse fields of research. I would also like to welcome the speaker for today's talk, Dr. Anita Pandey Ji, Professor of Biotechnology at Graphic Era deemed to be University, Dehradun, Uttarakhand. She initiated her career as Scientist Fellow at CSIR IHBT Palampur in 1992. She joined as scientist at GBPAL National Institute of Himalayan Environment, Almora, Uttarakhand, an autonomous institute under Ministry of Environment, Forest and Climate Change. Her major research areas have been extremophiles, rhizospheres, biodegradation, fermented food, water microbes, and antimicrobials. Having participated in several exchange programs, workshops, and conferences to share her knowledge on both national and international forums, she has 150 papers to her credit. Also recently published a book entitled Unique Ecosystems, Amazing Microbes under the series Extreme Environment. Dr. Pandey is associated with several scientific societies such as the Association of Microbiologists of India, Microbiologist Society of India, World Federation of Culture Collection, and many more. With this brief insight, that's all from my side. Now, without further ado, over to you, ma'am.
Welcome, Dr. Anita.
Yes, shall I start?
Yes, you can start.
Okay, good evening to all. I am Anita Pandey from Department of Biotechnology, Graphic Era deemed to be University. At the outset, I would like to congratulate MBSI for its multifarious activities and for its expansion in different states of India under the leadership of President MBSI, Professor Arvind Deshmukh, and Dr. Saranjit Singh, President MBSI for Chandigarh, Leh, Nagaland, and Manipur. I also thank Dr. Saranjit Singh for having me invited to share my experience on the microbial diversity, with particular reference to Indian Himalayan region. In fact, Dr. Swaranjit has been a very precious collaborator, particularly for accessioning the novel microbial cultures in MTCC IMTech of Himalayan origin, and then collaborating in a 2 weeks workshop, Exploration of Microbial Diversity, a Polyphasic Approach, that was organized by MTCC in 2002. And then he was our guest speaker in a workshop, Microbial Diversity, Isolation and Characterization, that we organized in October 2007, while I was working at GB Pant National Institute of Himalayan Environment, Almora. I'm also happy to share that our senior professor, Dr. Nishant Rai from Biotechnology Department, is now President MBSI Uttarakhand. In this talk, first I'll give a brief introduction of mountains in general, Himalaya in particular, and then I will share my experience majorly on extremophiles and rhizosphere microbiology. Just a minute. So these are my past and present affiliations. I started my career from CSI laboratory, and it is now known as Institute of Himalayan Bioresource Technology, Balampur, in Himachal Pradesh. From there, I moved to GB Pant National Institute of Himalayan Environment, Almora, Uttarakhand, and this is a autonomous institute under Ministry of Environment, Forest and Climate Change.
Development.
I was also visiting scientist at USAT, Karlsruhe, and presently I am with Graphikira, deemed to be university, a very renowned and highly ranked university in Dehradun, Uttarakhand. My research areas have been extremophilic microorganisms, rhizosphere microbiology, antimicrobials, fermented foods, and water microbes. The purpose of this I discussed with Professor Swaranjit Singh, and the purpose of this talk is to develop an understanding on the microbial diversity of Indian Himalayan region, right from ecology to biotechnology. In general, mountains are resources of global significance. This includes biochemical resources, genetic resources, and biodiversity and niche products. Mountains provide water, food, they provide hydroelectricity, they provide timber, they provide fiber, and mountains are the sites for recreation and ecotourism. Year 2022 was observed as International Year of Sustainable Development of Mountains, and mountains represent the unique areas for assessing the climate change impacts. And these days, in this background, mountains are being studied for the rich biodiversity and their adaptations in the present climate change scenario. In Indian context, we have Himalaya, and Himalaya includes the highest and the youngest mountains in the world, a major challenge to the mountaineers throughout the world. In Himalaya, we have more than 110 peaks rising to an elevation of 24,000 feet above sea level. This is like in Himalaya is shared by 8 countries, and these countries alphabetically are Afghanistan, Bangladesh, Bhutan, China, India, Myanmar, Nepal, and Pakistan. And the source of this map is ICMOD, International Center for Integrated Mountain Development, Nepal, Kathmandu. In India, the IHR extends to, or Indian Himalayan region extends to 11 states. Out of these 11 states, we have 9 fully and 2 partially states. In fully Himalayan states, we have, again alphabetically, Arunachal Pradesh, Himachal Pradesh, Manipur, Meghalaya, Mizoram, Nagaland, Sikkim, Tripura, and Uttarakhand. In Assam, we have 3 hill districts, and in West Bengal, we have 2 hill districts. Besides, we have 2 union territories, Jammu and Kashmir and Ladakh. This is a beautiful sight in Leh. We can see the peaks and ranges in Himalaya, and also the human settlements in the valley. Biodiversity, the Himalaya represents one of the 36 globally recognized biodiversity hotspots due to its unique and rich biodiversity. And in biodiversity, there are 3 major components: animal diversity, plant diversity, and microbial diversity. While animals and plants are visible, microbial diversity or the microorganisms are not visible. They are too small in size, they are microscopic, and they need magnification. However, microorganisms are an integral component of each and every ecosystem, as they are ubiquitous in nature. And they are ubiquitous because of their small size, their easy dispersal. Microorganisms are metabolically versatile and flexible, and they can utilize a wide range of substrates as nutrient source. They possess ability to survive and multiply in diverse habitats, including anaerobic. For example, Clostridium is an anaerobic microorganism, and most remarkable thing is they can colonize extreme environments where plants do not survive, where animals do not survive. Now, Indian Himalayan region presents great variation, particularly with respect to its topography, its geography, the soil factors, the edaphic factors, and the climatic conditions. And these variations in turn support a variety of habitats, including extreme environments. Microorganisms have applications, ecological as well as biotechnological. They provide ecosystem services. They contribute in nutrient cycling, for example, carbon cycle, nitrogen cycle, phosphorus cycle, and they are also contributing to biodegradation. Microorganisms are important in medicine. We get a lot of antibiotics, such as penicillium and streptomyces. Then microorganisms are important in industry. For example, the baker's yeast is a $1 billion industry, and this is produced by Saccharomyces. Microorganisms are important in agriculture. We have nitrogen fixing microorganisms, we have phosphorus solubilization microorganisms, and these microorganisms can be prepared, can be developed in form of biological fertilizers, and we can minimize the use of chemical fertilizers in our agriculture fields. Fermentation is a microbial process, and we have a variety of foods and beverages. In fact, these days, we are quite familiar with probiotic foods. Microorganisms are also used as biotechnological tools in genetic engineering. For example, E. coli or Escherichia coli, it is also used in transformation studies. Then we have agrobacterium, which is used in hairy root cultures. We have Thermus aquaticus, which is the basis of our PCR, polymerase chain reaction. For bioprospection, this is the approach. There are 3 major components in bioprospection of microbial communities: diversity, applications, and preservation. In diversity, we study a variety of microorganisms. For example, bacteria, actinobacteria, fungi, yeasts, microalgae, etc. Then in second component is the applications. Here we perform a lot of screenings. So screenings for biomolecules like antimicrobials, enzymes, and pigments. The first plate is a soil dilution petri plate where we can see a lot of antagonistic microorganisms. All these are plates from high altitude soil samples. The third component is in bioprospection of microorganisms is the preservation, and which I suppose is the most important. Usually in every laboratory, we have a culture collection with a code number, but it's important to accession these cultures in national or international repository. In India, we have 3 international repositories. We have MTCC, Microbial Type Culture Collection, Institute of Microbial Technology, Chandigarh. This is a CSIR organization. Then NCMR, National Center for Microbial Resources, at National Center for Cell Science, Pune. This is a DBT organization. And then we have NBAIM, National Bureau of Agriculturally Important Microorganisms, MAU. This is in Uttar Pradesh, and this is an ICR organization. Now I'll come to extremophiles and plant microbe associations. My major experience comes from these 2 areas. Extremophiles are the microorganisms that thrive under extreme environments, from geothermal areas to polar regions. Whenever there is a climatic event or a geological event, microorganisms are the least affected life forms, or we can say they are the best survivors. And this is by the virtue of production of enzymes, which are referred as extremoenzymes. These are various classes of extremophiles. On the basis of temperature, we have psychrophilic microorganisms, we have mesophilic, thermophilic, and hypothermophilic microorganisms. On the basis of salt, we have halophilic microorganism. Then for high pressure, we have barophilic microorganisms. With respect to pH tolerance, we have acidophilic and alkalophilic microorganisms. Temperature is one of the most important factors in high altitude areas. And this is classification of microorganisms on the basis of temperature. We have psychrophiles that they will grow up to 20 degrees Celsius. Between 20 to 40 degrees Celsius, we will have mesophilic. Above 40 until 60 degrees Celsius, we will have moderate thermophiles. Then above 60 degrees Celsius, we will have extreme thermophiles. Then up to 100 degrees Celsius, we will have hypothermophiles. So first I will talk to the thermophilic microorganisms. We have hot springs, which are the manifestations of geological activity. These were 2 of our study sites. Both are in Garhwal region, district Chamoli, and this area is referred as Tapovan. Soldhar and Ringighat, these were our 2 selected hot springs. In Soldhar, the temperature is as high as boiling, 95 degrees Celsius, where the altitude is 1900 meters above sea level. Ringighat is located at 1850 meters above sea level, and here temperature is relatively low, that is 83 degrees Celsius. These sites are oligotrophic and very poor in terms of nutrition, slightly alkaline in nature. Here, in case of thermophilic microorganisms, the major challenge is the isolation. And here we use a range of media, different dilutions, different growth conditions, such as a range of temperature, a range of pH. We can also use autoclave samples for plating to get thermophilic microorganisms. This is quite interesting. We can see this is a bacteria that was identified as Geobacillus stereothermophilus. And here we can see in picture 1, there is a terminal end has a bulbous structure. This works as a food reservoir. And when we maintain this culture in the laboratory, there is reduction in size because it's no longer required. And then the third one, we can see this is after autoclave twice. Third time it will not grow. So this shows the resilience of these bacteria. We could also isolate another remarkable microorganisms. For example, the filamentous organism. We couldn't identify them because they were under VBNC, viable but not culturable state. Then we had a variety of bacilli. So these microorganisms were identified as species of Geobacillus, Bacillus, and Paenibacillus. They are ecologically important. They produce enzymes like amylase, which are important for their resilience, and also they're very useful in the industries. These sites are also colonized by cyanobacteria. At 95 degrees Celsius, we have few cyanobacteria or the algae, while at 83 degrees Celsius, there were several, like we could isolate 37 species represented by 17 genera. Many of them contribute to biological nitrogen fixation. These sites, they experience a number of different kind of disturbances. For example, the natural calamities such as landslides, etc. Then we have anthropogenic activities. We can see the road construction work in at these sites. And this particular Soldhar hot spring is in Nanda Devi Biosphere Reserve. And here we can understand the importance of microbial culture collection. All these cultures are available in depositories. Another extreme is low temperature environments where we have high mountains, alpines, cold deserts, glaciers, and these sites are colonized by psychrophiles or psychrotrophs. This is just an example. Himalayan glacier in Uttarakhand area, the Pindari glacier, and this is a snow-clad Himalaya. These have been some of our sites for isolation of microorganisms. These low temperature environments are dominated by species of Bacillus. We could isolate more than 25 species of Bacillus. That it is mainly because of their endospore forming nature. Next to Bacillus, we have Pseudomonas species. We isolated more than 50, and they are ecologically important. They are biotechnologically or industrially important. They produce a range of antimicrobial substances, many bioactive compounds. We also have rich diversity of actinobacteria in these sites. And actinobacteria, it's a class between bacteria and fungi. Streptomyces are the most common, which are best known for the production of antimicrobials. They produce lytic enzymes like amylases, proteases, lipases, chitinases. And interestingly, we also have a variety of yeasts, cyclotolerant yeast in these areas. Now we have a range of cold adapted fungi. We could isolate more than 35 species of penicillium, and they were identified on the basis of molecular characters. Next to penicillium, other dominant fungi were Aspergillus, Trichoderma, Bacillomyces, Cardosporium, etc. So the important thing is, the point is, these sites are dominated by Ascomycota, or we can say the cold adapted Ascomycota. And this is mainly because of their spore shooting nature. They have elevated sporulation. If we grow these microorganisms, these fungi at optimum temperature, we will get moderate mycelium with moderate sporulation within a week. But when we grow these fungi at low temperature, they will show heavy sporulation with minimum mycelial growth on prolonged incubation. And they grow with a very peculiar growth pattern. And these characters, the spore shooting characters, is important in adaptation, and it is also important in industry. These microorganisms produce a lot of pigments, like this is penicillium. We get different pigments at different temperature, and this is an important character for their own survival. On the other hand, these pigments have industrial applications. This is another example, like we have a cytotolerant bacteria, Serratia marcescens, and it produces cold active pigment, the producin, and we can see at 14 degrees Celsius, it is producing this red pigment. At 24, the pigment is absent. So this is a character again for ecological resilience as well as biotechnological applications. This bacteria, Serratia, it also produces lacase, and by the virtue of this lacase, it contributes to biodegradation. These fungi, the Ascomycota, they also contribute to biodegradation, and biodegradation is a slow process under low temperature environments. They produce a number of enzymes like lignin peroxidase, manganese peroxidase, and lacase. And the production of these enzymes is relatively slow, but for a longer period. This is a very unique characteristics, like on the basis of pH, we divide microorganisms as acidophiles, neutrophiles, and alkaliphiles. But these microorganisms, be it a bacteria, be it a fungi, thermophiles, or psychrophiles, they can tolerate 0.5 to 14, a very wide range of pH. And again, this is ecological important character, and this also have applications in industry or biotechnology. Now, in low temperature environment, we have a plethora of vegetation, and these sites are important for studying plant microbe associations, their adaptations, and microbial indicators. In Himalayan region, in high altitudes, we experience heavy rainfall as well as snowfall. And these sites are important for studying rhizosphere effect, plant microbe associations, bioactive compounds, enzymes, phosphate solubilization, biological nitrogen fixation, and microbial indicators. These are some of the important species of high altitude. We have species of Quercus, we have abies, rhododendrons, Texas, pine, etc. This is a very important, very interesting observation. Usually, if we talk of a rhizosphere effect, which can be calculated in terms of RS value, rhizosphere to soil. Soil means non-rhizosphere value. We divide rhizosphere microbial counts by soil non-rhizosphere counts. And usually, in rhizosphere, we are likely to have higher microbial populations. And here we can see that there are some species which are from alpine regions, like Abies pinto, Betula utilis, Rhododendron campanulatum. Here we have RS ratio well below 1, showing the negative rhizosphere effect. And these sites are very important sites for isolation of antagonistic microorganisms. Another interesting thing is, like wherever we have this negative rhizosphere effect, we also have, we noticed, we observed, we recorded the lowering in soil pH. For example, if we see in Abies pinto, in rhizosphere, we have 5.3, 4.8, 4.7 pH, while in its non-rhizosphere, we have 6.3, 5.5, 5.6 pH. Similarly, in case of Betula utilis and Rhododendron campanulatum. And again, these sites are important, suitable sites for isolation of acidophilic microorganisms. These are the applied aspects of rhizosphere. In 1904, German scientist Hippner, he coined the term rhizosphere, and he also explained the two important aspects of rhizosphere. Number one, the plant growth promotion. Number two, the biocontrol or the disease control. And now the rhizosphere research is receiving much attention for developing biological fertilizers. And there is a global preference for organic or environment-friendly products instead of chemical fertilizers. Usually chemical fertilizers are preferred as source of nutrition, mainly because they are readily available, they are easily handleable, we can have predictable yield. But there are documents that these chemical fertilizer, use of chemical fertilizers is not good. It is harmful for the ecology, overall ecology, the soil health, even the water resources are contaminated. And there are many failures in the use of these rhizosphere microorganisms as microbial fertilizers. And the reason is, in most of the cases, the microorganisms are picked up from one area and introduced in another area, while microorganisms are environment specific. So we need native microorganisms. For example, I worked in Himalayan region, so all our isolations were from high altitudes, and then we go for their identification, then characterization, particularly for their traits related to plant growth promotion. Then we go for bioformulation, and then field experiments, field trials we will go for. And this is the very systematic approach. There are two major aspects. One is ecological competence and another is rhizosphere competence. And in this background, endophytes will be the most important organisms which should be developed in form of microbial fertilizers. Endophytes refer to the microorganisms that inhabit inner tissues of higher plants, at least for one period of their life cycle, without causing any apparent systems. And this is likely to be a symbiotic association where plant will provide nutrition as well as the shelter to the endophytic microorganisms. In turn, the microbial partner will provide a plethora of bioactive metabolites that contributes toward the plant growth as well as the competitiveness of the host in a given ecosystem. This is an example for isolation, and we have lentil. Lentil is grown in high altitudes, and you can see the infected roots, and this can be isolated. This was trichoderma, and we identified as trichoderma gemsi, and it's a good phosphate solubilizer. The species also possess inhibition property against a range of phytopathogens. So this is a suitable organisms to be developed in form of bioformulation. Now I will talk about some important associations in high altitude plants, because rhododendron is very important. It was considered as an indicator of climate change, mainly because of its early flowering. Here I want your attention on E, slide picture number E, where we have dark septate mycelium, dark septate endophyte. It's not that we didn't provide a stain to it. These are the structures, they don't take any stain, mainly because they always appear as dark brown or black color. And this is mainly because they are rich in melanin content. This is another example from high altitude wheat. Wheat is also grown in high altitude, and it was a climate change related project. We worked on 3 altitudes in high altitude region of Uttarakhand. And this was a very important, remarkable observation. At highest altitude, that was 2,400 meters, we observed these dark septed endophytes. And the important thing is, at this site, the crop remains under snow for more than 1.5 months. So the crop is resilient, and the microbes they are harboring also resilient to climate. We also found these structures in high altitude Himalayan trees like Himalayan birch or the betula, then pine, then oak, then Texas. I proposed this hypothesis. This was an opinion paper. Can we consider these dark septed endophytes as indicators of climate in mountain ecosystems? And now at Graphicira, I'm working on this hypothesis using a mid-Himalayan tree, that is Diplopnema, and a high-altitude tree, that is Abies pintro. Some example where we used endophytic microorganisms in propagation and conservation of some endangered plant species. This is Ginkgo biloba, and this is referred as a living fossil, being the only living member of Ginkgoaceae. And this is known for its resistance to diseases and longevity. It has several medicinal properties. It produces the compounds like flavonoids, ginkgolides, and it's known as a very slow growing tree with ecological importance in pollution control. Botanical Survey of India referred this species as rare. Here we isolated endophytic bacteria from the cortical cells, the mycorrhizal roots, and the bacteria we identified as a species of Pseudomonas. It is mycoid one. We developed a bioformulation, a liquid bioformulation, and we used stem cuttings for inoculation. This we worked in collaboration with the forest department, and we provided hundreds of plants of Ginkgo biloba to be planted in Kalika nursery in Ranikhet. Another very important tree of high altitude, this is Texas wallichiana. In English, it is known as Himalayan yew. In Hindi, Thuner, and it belongs to family Taxaceae. This is also mentioned in IUCN Red List. And this is very well known for its active ingredient, that is Taxol, which is very well known for its anti-cancerous property. The roots were found rich in penicillium and aspergillus colonization. We could also isolate endophytic bacteria from the roots of this Texas wallichiana, and they were found positive for plant growth promotion and biocontrol characters. Interestingly, these organisms were also studied for their ability to produce Taxol. For propagation, we used a consortium from roots of these plants, and again, this we did with collaboration with the forest department, and we provided again hundreds of plants to be planted, to be transferred in different locations in Jageshwar area and Ranikhet area in Uttarakhand. I have good experience on rhizosphere microbiology of tea, and for this I worked across the Himalaya. We have centers in different states, distant Himalayan states. In Himachal Pradesh, like we have this Banuri Tea Experimental Garden. Then in Kosani, that is in Uttarakhand. Then we have in Sikkim, Temi Tea State, where we have a center. Then we have this Darjeeling. And you may not be knowing that tea in general, it grows in form of a medium tree, but this is probably the only plant which is maintained as a shrub or as a bush by continuous pruning cycles. And this was the first plant that was reported for its negative rhizosphere effect from our laboratory. Bacteria will be the most suppressed group in the established tea rhizosphere. Actinobacteria and fungi, they were also suppressed, but to a lesser extent. Tea roots favor the colonization of a specialized microflora which is dominated by the antagonistic species. In general, it is mentioned that tea is grown in acidic soils, but it is also a fact that it is the tea plant that makes the soil acidic. This is the complete association. Spore comes in contact with the tea roots and then penetrates, then development of various structures, arbuscules, vesicles, etc. And very beautiful site. I have visited many tea gardens, and this is in Sikkim. Women work in these gardens, and these are just beautiful sites. This is also very interesting, an example of endophyte. And this association is among 3 partners. Like in our previous institute, in my previous institute, we had a salix patch. And every monsoon, we will get these puffy balls, the mushrooms. And inside these mushrooms, we could isolate fungus that was identified as Itupela, and it produces lacase. So it contributes to biodegradation. We are likely to have many such examples in high altitude. So the endophytes, they are likely to produce bioactive metabolites, similar the metabolites that are produced by their host plants, and these endophytes certainly contribute to the adaptation and host competitiveness under low temperature environments. Some other areas which are very important, like aquatic microorganisms, Uttarakhand or the Himalayan states, they are home for Himalayan rivers. And this is a site in Uttarakhand. This place is called Jageshwar, where we have Deodar forest. We have about 120 temples in a very small region. Then we have a river flowing there, that is Jataganga. And this is used for many purposes, anthropogenic purposes. Then we have festivals. This is under Archaeological Survey of India. And these sites are important for studying the biological indicators such as coliforms, then pathogens, and monitoring of water sources will be a very important aspects for which we have Bureau of Indian Standards. Also, I have some experience on traditional fermented foods, and these foods are now receiving preference as important source of natural probiotics. We have many such practices in Himalayan region, especially in the state of Uttarakhand. And here you can see, this is an example of yeast bacteria fermentation. And there are communities which make different products. They have specialized utensils for that. And they can prepare a beer kind of a drink, then alcoholic drink, and also some semi-fermented food like snacks. So this was all for this presentation, and it was possible only because of my scholars. They worked several theses, several PhD programs, dissertation programs were done on these areas. And most of my scholars are working in nationally, internationally renowned organizations. We also had senior professors from different countries who came to my lab to work on rhizosphere or extremophiles. I got support, financial support from various organizations, mainly from MOEF and climate change. Then NMHS program is there, from DBT, DST, CSIR, ICMR. FICCI supports this African program, then UCOST and USER in the state. This is the Great Himalayan National Park, and this was recognized as World Heritage Site for Biodiversity by UNESCO in 2014. And these are the sites. Many such similar sites are there in Himalayan region, unexplored for microbial diversity. So we certainly need to work on these areas. Thank you very much once again. Thank you so much Dr. Suranjit Singh, and all the participants. Thank you.
Thank you very much, Dr. Anita.
Yes.
Your talk was excellent.
Thank you so much, sir. Thank you
You-
so much for the opportunity.
You
I will be happy to, if there are some queries. Otherwise, my email is already there. Anybody, if somebody wants some discussion, they can write to me. Yeah.
I just want to say one more thing. Your talk was about diverse environment.
Right.
It was a great talk. I really enjoyed your talk.
Thank you so much, sir.
Now we can have questions. I can see Nishant. I can see Dr. Bhagyaraj.
Dr. Nishant is our MBS.
I know.
President. Yeah.
He's a president of MBSI.
Yes, he is our
UK.
colleague, senior professor. Yes.
He's your colleague. Very good.
Matter of proud for us. Yes.
I know him very well. Huh?
Oh, okay, good.
I know him very well. Yes.
This I didn't know.
And
Yes. Professor Bhagyaraj is like my mentor, my teacher. He has been always there for me. Yeah. Right from my
Yes.
Thank you. Thank you very much, Dr. Anita. So nice to see you and hear your
Yes,
talk.
yes.
Listen to your talk.
Yes, thank you so much, sir. Sir, I have learned so much from you, sir. Thank you so much.
Yeah.
Yeah, yes.
You can ask something, sir.
Okay, I'll just add a small doubt. The thing is, the organic matter decomposition by the lignocellulosic microorganisms, because of the production of enzyme is very slow, it takes a longer time. That's what Dr. Anita said. How long does it take for the decomposition of organic matter under such low temperatures?
Sir, exactly I cannot say, but it continues. The activity continues up to 45 days, 50 days, like that.
Oh, more than that. Yeah, it takes longer time.
The enzyme production is at lower side, but for a longer. Quantity will be less, but it's for much longer under laboratory condition.
Okay. And one more thing I just wanted to ask regarding the rhododendron.
Yes, sir.
Yeah, you described the fungus associated with it. Did
Yes.
you also come across ericoid mycorrhizal fungi?
Sir, I was not expert on that.
Because it is supposed to be colonized by
Earlier
ericoid.
also you talked about it, sir. But whatever fungi, initially I was a bacteriologist, but I got interested. I was not a mycorrhiza expert, but slowly I learned. But this endophyte dark septate, it's very interesting, sir, because sir, it replaces mycorrhiza with altitude.
Okay. Yeah.
At high altitude. Right now we have two species. I have two students in graphic era. One is working on Diplocnema, that is in mid-Himalayan. We don't have any dark septate. But the one Abyss Pindro, it's on high altitude. Alpine, there we have these structures. So I'm focusing on these organisms these days, sir.
Okay. Thank you very much. Yeah.
Okay, Dr. Dharam Singh, please.
Okay, sir. Can you see me?
I'm
Yes.
so happy to see you, Dr. Dharam Singh.
Thank you so much, ma'am. It was indeed a privilege to listen your talk, and Dr. Sanjeev also. And this
The thing is,
someone
I
actually.
started my career from your institute.
Yeah. I know you. I know you actually for a very longer time period, but we never actually met personally. Yeah. So thank you very much for your this excellent talk, and I can see actually it's blend of rich experience and a thorough experimental your experience in this area. So and since I also work into this area, and I do belong to this Himalayan route also, and my
students, our dissertation students are always there in your institute. So we plan some collaboration. Okay.
Yeah. Many students actually.
Yes, I know that.
They have undergone for 6 months.
And we now have a MOU with your institute.
Okay.
Last year only when Sanjay was there, we signed MOU.
Right. So my lab focuses on the industrial enzymes on Himalayan region.
I have seen your papers on novel organisms are also there.
Right.
That's great, sir.
And another is the bioplastic area that I am working on. So these are to my specific area. So I have during this period actually it's a lot of challenge in working with the Himalayan microbes, starting from this sample collection to the going for their characterization to then functional attributes. So
sampling is the most important, sir.
Yeah.
Sampling
So what I the difficulty that I face in the enzyme bioprospection from the Himalayan microbe is we get a lot of inclusion bodies when we express into the system. So I work with the different enzymes, and I feel that for the enzymes from the Himalayan microbes, because they are extremophilic and normal expression host and then the vector system, they are not more stable. So maybe the specific expression host from the Himalayan microbe, which can be actually a model organism that can be created. So that may be actually, that's what I feel actually. Otherwise, we have a lot of challenges.
One thing is there, like in high altitudes, please take care of yeasts. I couldn't do much, but I was getting a lot of yeasts. I was having limitations, like not many people were there. So, but yeast is very important. We have Rhodococcus, Rhodotorula, many yeasts are there in high altitude. So they can be, you can take care of that. Their resilience, how they are adapting, how they are surviving in low temperature environments. And I always consider resilience to applications. Adaptations and the application part. So now you are there, you are young, so you can take care of that.
Yes. Another question is
actinobacteria, high altitudes.
Yeah.
There
are we get actually a lot of those actinobacteria.
The actinobacteria will be there, but their maintenance is little difficult. Very fast they require subculturing.
Right. So another question is related to your expertise when you study this plant microbe interaction, and a lot of you have found a lot of endophytes. So when you actually do in vitro cultures, so did you find actually after many generation of subculturing, they still maintain that functional attributes, or they get lost with the subculturing?
Yeah, but if you are maintaining the cultures in this log stock, this problem I think can be minimized.
Okay.
Fresh culture is always a fresh culture in terms of activity.
Okay, I have one question, madam.
Yeah.
In Leh and Ladakh,
yes,
there is very low concentration of oxygen.
Okay.
And then there is very high UV falling on Leh and Ladakh. So low concentration of oxygen and high UV falling, does it make some changes in the biodiversity?
We will have to, we have to plan some project on this line, some anaerobic isolation of anaerobic microorganisms.
Right.
Then we can answer all these things. The youngsters, I think you are associated with Ladakh.
Yes.
Our center is also there. We have a center there, but I don't think there is any microbiologist. But I will find out if there is. So it can be planned until.
Let us plan that study. It is very, very good study.
Yes, sir. Actually, in my study, with respect to your question,
Dr. Dharam Singh will take care of that.
Yes, I want to give a small reply to that one. What we found actually, because you are right, actually, it's a high UV radiation in those areas, and from my sampling site also, I go from 3,000 to the up to 5,500 meters height.
Okay.
What we found actually, as you go up to the higher altitude, though number of bacteria they getting decreased in number, but they are more pigmented, and this pigmented we have to check for they have a high and UV protected functions.
Right.
So they produce actually several kinds of pigments, and they really help in the UV protection. And with that, actually, their size, actually, the microbe size is getting extremely into the smaller size. So that is a very important area, and our observation. Yeah.
And one thing, they keep on mutating very fast because of UV. They keep on mutating every day.
Because of radiation.
Variation is there.
So they have actually low GC content because I did the genome, whole genome sequencing also. So that low GC content is the one of the features for the high mutation rates.
Okay.
So it was a real interesting talk today.
New areas to work, sir. Very interesting.
Yes.
Because I was restricted to temperature or pH, but the radiation thing is there. So many things. The UV thing. All these things are barophilic microorganisms and all that pressure.
Yes. You know, I went to
For
Leh,
next generations.
I went to Leh twice, Leh and
Yes,
Ladakh,
you told me
and
that.
I got soil from there.
Yes.
And in IMTech, we did the metagenomic library.
Okay.
And
good.
every time I went, the
Mhm.
library was different
Oh,
because the rate of mutation is very, very high,
it's very important.
very important.
Report it fast.
Other they also produce exopolysaccharides, a lot of
Yes.
exopolysaccharides.
Yes, they do that.
Yes.
And they have metalloenzymes,
Yeah.
metalloenzymes like iron and calcium. They
Yes.
have selenium enzymes are heavy to protect against the cold.
Yeah, most important aspect will be the adaptation strategies.
Yes.
That is molecular
Yes.
level, biochemical level, morphological level.
Right. We'll discuss about this. All three of us will discuss.
Thank you very much.
So
Thank
if
you.
there is no more, if there is no more
I
question,
knew your name.
if there
Yes.
is no more question, then we can conclude the session.
Okay, good.
And thank you, madam, once again for the nice talk.
And thank you
now-
for giving me this opportunity.
No, you
It's nice to see you
enlightened
after
us,
so long time. It's nice to see Professor Bhagyaraj, who has been like a teacher for me.
Yes.
Dr. Karan Singh, for the first time I'm interacting with him.
Yeah, nice to see you and listen to your talk. Thank you. Thank you very
Thank you,
much.
sir.
Very
Thank
nice.
you very much, sir.
Supriti, you can please do the vote of thanks.
Yes, sir. Thank you for such knowledgeable talk today, ma'am. Before we conclude this call, I must mention our deep sense of gratitude to the President of Central Council of MBSI, Dr. Arvind Deshmukhji. Thank you, sir. I would like to acknowledge my sincere words to our respected President of Tri-City, Dr. Saranjit Singh Ji, who is acting as an energy source. Thank you, sir. And I'm happy to mention my obligation to the most valuable speaker of today's talk, Dr. Anita Pandey, for her valuable words. Thank you, ma'am.
Thank you. Thank you, everyone. Thank you so much.
Thank you. Thank you, ma'am.
Thank you. Thank you, Supriti, for the nice welcome and the vote of thanks. Thank you very much.
Thank you, ma'am, for such a valuable talk, and indeed it was a good presentation.
Thank you.
Good.